All right, so we're moving on to the nervous system, the control system for our body. And let's move on into this, the brain, the spinal cord, the nerves. So start out with the gross anatomy of the system. You see that it really comes down to these two divisions. There's what's called a central nervous system and just two organs, the CNS, common abbreviation. So we have the location for the brain and the spinal cord, right, inside of our skull, inside of our vertebrae. And now everything else, all the remainder of the nervous system, and it's truly the smaller percentage, the smaller remaining fraction, is called peripheral nervous system, PNS. So this is all nervous tissue outside of the brain and spinal cord. That's a very simple description. So what's included? Well, nerves that are, say, on your arms and legs, the receptors we learn, for example, in the skin, a tactile corpuscle. And you see the term here, neuroglia, not to be confused. There's certain neuroglia that are only in the PNS, but there's also some neuroglia that's only located in the CNS. So as far as the anatomy, that's the simple breakdown, central nervous system versus peripheral nervous system. But altogether, a very simple description of how the nervous system functions is, this is the control system for our body. So we're first able to detect stimulation in the environment. We could see light, we could feel pain and pressure, right? We could feel temperature. And then now we could respond to that stimulation. And we are able to have these very rapid, fast responses to that incoming stimulation. And the way that our nervous system controls these responses is by the second bullet point. We're able to stimulate the activities of other organ systems. So a really simple example, I do this all the time. You know, I'm cooking or something, I touch something hot, and I'm feeling that stimulation, something's hot. If it's hot enough, it'll be painful. And then I get that quick reflex to pull my hand away. Well, actually pulling my hand away comes from, of course, the nervous system controlling my muscles. So a simple example of withdrawal reflex. You detect stimulation, you interpret that stimulation, and then very rapidly you have a controlled response. So taking a look at the function of the nervous system, here's a great figure in the textbook. And we're going to start by going over just these terms here, what's called sensory, and another word for sensory, a synonym, afferent. And then the opposite, you see here, motor or efferent. So these are, of course, describing the direction in which information travels. So a sensation or afferent signal, you see this travels from the PNS, think of the various nerves around your arms and legs, and sensory or afferent information is destined towards the central nervous system. So we can become aware, interpret our senses. Motor or efferent's the opposite. So motor signaling could originate in the central nervous system, and then it goes out into the PNS to control various organs around our body. So sensory signals, again, sensation is another term for afferent, going towards the brain and spinal cord. Motor efferent, that's now the command, the signaling from the brain and spinal cord going to control various organs around our body. So sensory versus motor. So a similar diagram is like so. I'm just spelling that out. So afferent or sensory, afferent and motor. And you notice these terms apply to both central nervous system and peripheral nervous system. So starting there, here's a little mnemonic. SAME, sensory and afferent have the same meaning. So signals going towards the brain so we can perceive our sensations. Motor and efferent are synonyms, and you see signals traveling away from the brain out to control some organs. So always sensory and afferent. We're signaling towards the brain, towards the CNS. Motor efferent, signaling away from the brain and spinal cord to control some organ. So it's the direction. So we now see within sensory, there's two types. There's somatic sensory and visceral sensory. We're not worried about this one yet. But that also applies to the motor portion. So there's the somatic portion here on the motor division. And this term autonomic, this is another word for visceral motor. So here we just want to point out there's a visceral sensory, somatic sensory, somatic motor, and autonomic is truly just another name for visceral motor. And you see those four labeled here. So let's describe each of these. So to begin with the sensory side, well, sensory always means the signal is traveling towards the brain and spinal cord. So sensations. The difference now, a somatic sensation, this is some type of stimulation in the more superficial parts of your body. So you want to think about, you know, something, a stretch, a cramp in your muscle, something's touching, irritating your skin. So more superficial sensations, that's a somatic sensation. And those irritations on your skin or muscle or temperature, ultimately that somatic sensory information is ultimately destined towards the brain, and you become aware of that. A visceral sensation, these are once again sensations, but these happen and occur deeper in your body. So you want to think of organs that are in your torso. So your stomach, your intestines. So think about, you know, after you eat a meal and your stomach is full, that sensation. Your digestive organs are a perfect example. The pressure in your lungs, any perception in your heart, like a heart attack, the death of cells, that would be a visceral pain sensation. Okay, so we see it's the location. Where do we interpret these senses at? And we could go back here. And again, you notice here, visceral sensory receptors, internal organs, cardiovascular, respiratory, digestive, all great examples. Sensations in your deep internal organs. Somatic sensory receptors, again, great example. Stretch in your muscle, any type of stimulation, pressure, touch on your skin. So a more superficial location, but they're both senses. Whereas now if we go to the motor side, so there's somatic motor, and here's where these alternate names come in. So autonomic nervous system, another name for that is visceral motor. So two names for the same thing. Somatic motor is when you have direct conscious control of skeletal muscle. So this is the only type of tissue that has somatic motor control. So what this means, if you want to wave your hand, just simply thinking about the activity, you can just at will, anytime, wave your hand back and forward. So that's what the meaning of somatic motor control is, just the conscious control of your skeletal muscles. Visceral motor or autonomic nervous system, well, the opposite here of conscious, this is now this involuntary or unconscious level of control. So the example here, of course, would be the motor function to keep your heart beating, smooth muscle that's in your arteries or veins, or any type of glands. So the glands, this would be a good example. You could just simply smell food or see food, and you could start to produce saliva. It's not something you directly have to think about. Or your heart rate, right? You can't directly decide that you just want your heart to beat faster, but based on your mood and behavior and activity, you're going to have that unconscious or involuntary level of motor or efferent function. If we just go back to here, you see that somatic, again, motor. Somatic motor only applies to control the skeletal muscle. Visceral motor, we will point out, has these two separate divisions. It's autonomous function is really what the word means, autonomous, so without your direct conscious thought. And this is the only way that we could control smooth muscle, cardiac muscle in your heart, or any type of glands. So before we move on from this major topic here, quickly to point out the autonomic or visceral motor function. Again, that's another name for it. It has these two typical divisions that we'll talk about for now. There is a third we won't worry about, but the general two, sympathetic and parasympathetic. So these are just two different parts of this larger visceral motor, this involuntary motor function pathway. So now moving along, we have a nice section in the textbook that goes over just basic terminology for the nervous system. So you probably heard the words gray matter, white matter. So what we're looking at here, gray matter in your brain does have a darker color to it, and white matter actually does look white. So specifically the gray matter, you see the description, mostly, for the most part, large amounts of nerve cell bodies. So if I make a little sketch, here's my little nerve cell that I'm drawing. The cell body is this large portion right here, where I'm drawing the circle. So this entire cell is a nerve cell, this entire section, but just this circular part here is the cell body. And gray matter is just a large collection of principally nerve cell bodies. White matter is now principally myelinated axons. So going back to our nerve cell here, just this long, slender part here only, that is the axon. But what's meant by myelinated, this is when the axon is wrapped. So there's another type of cell that's literally wrapped around the axon to make it myelinated, and it gives that white matter appearance. A nerve fiber is just another word for an axon. So one single axon, so that's right here. But now we have these two terms that we want to group together, because you see nerve and a tract are truly the same thing, a bundle of axons in both cases. So now the difference is, well, location, and that's where you see PNS, peripheral nervous system, bundles of axons are called nerves. So you can think of the nerves that travel up and down your arm, for example. Whereas a very similar structure, bundle of axons, but in the central nervous system, that's what the term a tract is. So it's based on location. And then you notice the same thing down here. Ganglion and nucleus, we'll group those together. So we're talking about, right up top, nerve cell bodies once again. So gray matter, the cell bodies of a neuron. And when we're in the peripheral nervous system, the more specific name for a cell body is a ganglion. So a ganglion is just simply a cell body in the PNS. A nucleus, this is a cell body if it's in the brain or in the spinal cord in the central nervous system. All right, so we want to keep the terms paired together. That'll kind of help you learn and see that difference based on location. So now we're going to move into the cytology, just the distinct cells. We actually saw this very same slide, very early in the semester. So when it comes to the cells in the nervous system, there's only two general categories. What's called neuroglia. These are the supporting, aiding cells of the nervous system, and we'll discuss 6 examples for neuroglial cells. We see that they are more common, so this 5-to-1 ratio, the more common neuroglial cells. Whereas in the neurons, these are now the cells that are actually sending the sensory signals towards the brain or sending motor signals away from the brain. So these are the cells that are truly transferring and processing information, sending sensory information, sending motor information, processing information in the brain and spinal cord. That comes from the neurons. And that's where we'll start. So here's now a nice illustration of a neuron. This is what I was trying to draw on the previous slide. So to see just some of the parts, on one end, the receiving end, there's the various dendrites. This is where information comes into the neuron. Here you can see that cell body portion, and it's defined by containing the nucleus. And then you can see it tapers down to this long, slender axon. And some axons can be very, very long. In fact, neurons are the longest types of cells in our body. So that long cable, like axon, and then branching at the end, we have the synaptic terminals. So what's unique about a neuron is that these cells could transmit an electrical current. So a voltage will start at the dendrites, and then that electrical current, you notice, travels in one direction only from the dendrites through the cell body axon towards the synaptic terminals. So it's only in that direction. It's not possible for the current to go in reverse. A key trait about mature neurons, a mature neuron does not have a centriole organelle. Those are lacking from mature neurons. So because of that reason, we cannot, for the most part, replace neurons in our body. So when a nerve cell is destroyed, for the most part, we cannot replace those. But the good news is they're in the billions. Another issue with this illustration here, this is showing a completely exposed neuron, which is not the case in life. Almost all of the nerve cells' surface would be covered by those supporting cells. And this is just a shortened form. Glial cell, or the full word would be neuroglia. So those more common supporting cells wrap all around the majority of the neuron surface. And here from this illustration, neuroglia are absent. So here's a more detailed look at a nerve cell. We're not worried about this for now, but you can see the dendrites, the cell body. It tapers down to an axon hillock, leading to an axon, synaptic terminals. And again, the electrical current travels one direction towards the synaptic terminals. And it's just described here in the text. And I put a big cross on it so you don't have to worry about studying the text. Again, the principal idea is that the signal starts at the dendrites and then it travels towards the synaptic terminals. This figure here is showing now different distinct types and shapes of neurons, but go ahead and just ignore this section altogether. So we want to finish here with the nerve cells. So again, from dendrites to synaptic terminals, the nerve cells send an electrical voltage from dendrites towards the synaptic terminals. However, the way that a neuron communicates to another cell, so between neurons and other cells, there's a release of a chemical. So to communicate from these synaptic terminals to this muscle cell, we already learned the chemical involved in terms of muscle contraction was the neurotransmitter acetylcholine, ACH, and we're just describing that again. So the way that we signal from a neuron to some other type of cell is by the release of a neurotransmitter. And one that we just studied from the muscle section, ACH, would be that perfect example. So from neuron to muscle, from neuron to neuron, from neuron to any other cell, it's usually a chemical neurotransmitter passing between cells. So now we'll move on to the supporting cells, the more common neuroglial cells. And the first thing that we want to point out is they're divided by location. So there's central nervous system, meaning brain and spinal cord, and then PNS, peripheral nervous system. These two are not in the brain or spinal cord. And this figure is great because based on location, these two cells, a Schwann cell and a satellite cell, are very similar to two of the cells on this side. So if we just take a look, Schwann cells, there it is, myelination, and oligodendrocytes, myelination. So these two cells principally have the same job, just in different areas of the nervous system. So we want to group those together. And the same goes here for satellite cells. You see regulating gas, nutrient levels around neurons. This is very similar to astrocyte functions, regulating ion, gas levels, whatnot. So the astrocytes and the satellite cells are similar, but just in different locations. So we want to keep that in mind. And that'll cut down on some of the information if you just group these together. So let's begin here. This is a nice illustration, kind of putting some things together. You can see the neurons here in the blue coloring, and the supporting neuroglial cells, they're seen here in the yellow coloring, these yellow cells. And you notice we're looking at the spinal cord, a part of the central nervous system. You can see that there's the white matter, the lighter portion, and here it is labeled, white matter. You notice the axon that has the myelination around it, making it appear white. And then the deeper gray matter on the spinal cord, and this is where you can see groups of nerve cell bodies. So just as we started with the difference, white matter, gray matter. But what we want to focus on specifically now is this astrocyte. And you notice the astrocyte is this intermediate cell between the blood vessels. You know, it's a capillary. The astrocytes directly interact with the capillaries, but the astrocytes on the other side also directly interact with the neurons. And that's really their major job. They're an intermediate step between the blood supply and the actual nerve cells. So taking a look, starting with the central nervous system, the 4 types of neuroglia that we're interested in. So we have these very common astrocytes. We do have some ability to repair injured neurons. We said earlier, if a nerve cell is destroyed, we lack the ability to just simply replace them and make new ones, but we do have some ability to repair injured neurons. The bigger part of the astrocyte's job is right here. We saw in that previous figure, the astrocytes are that intermediate step between the blood supply, the capillaries, and the actual neurons of the brain and spinal cord, the central nervous system. So having that intermediate between the capillaries and the neurons of the central nervous system, the astrocytes are regulating the amounts of nutrients, the amounts of oxygen, various chemicals that could ever enter into the central nervous system. And it's said to control and regulate what's this blood-brain barrier. So chemicals that are in our blood, some of them are simply blocked from ever entering into the central nervous system. Whereas other chemicals, oxygen and sugar, we need high levels of those chemicals entering into your brain and your central nervous system. So ensuring that certain features, certain chemicals of your blood do not get into the central nervous system, that's the purpose, the major purpose of these astrocytes. So there's a few areas of the brain that actually do not have a blood-brain barrier, and you see some examples listed here. And the reasoning here, it makes sense. These are all areas that need communication with the bloodstream. Your pineal gland, for example, produces a melatonin hormone, a hormone. Hormones, by definition, need to get into your bloodstream. So it makes sense that this is an area that does not have a blood-brain barrier. But for the most part, most areas of your brain do have these very common astrocytes, and it has that diffusion barrier. Whereas another type of cell, if you look at the description, it's kind of similar. Ependymal cells, they once again surround capillaries, but these are in just limited areas of the brain. And these ependymal cells, they interact with capillaries, and they filter our blood plasma. And filtered blood plasma is now known as cerebral spinal fluid. And this circulates in both the brain and the spinal cord. So the central nervous system, the brain and the spinal cord, has its own type of circulation. So let's just compare these two, okay? So this is a great figure from the brain chapter 16, but we're going to see the difference between these two types of cells. So there's the astrocytes and the ependymal cells. So let's begin here. The astrocytes, we said that blood-brain barrier, intermediate step between the capillaries with our blood supply and the actual neurons of our brain and spinal cord. So in order for various chemicals to enter into the central nervous system, they have to filter through the astrocyte before they reach the neurons. And that property there is now regulating the makeup of our interstitial fluid, regulating which chemicals actually come into contact with the neurons of a brain and spinal cord. So that's the astrocyte blood-brain barrier. Whereas over here, you see some more capillaries, and here they are lined by these pink cells representing ependymal cells. So the blood plasma circulating through, only the material that could filter and pass through the ependyma. Now this fluid is known as cerebral spinal fluid, and this fills the hollow spaces, the hollow cavities that are in your brain or in your spinal cord. So both of these two types of neuroglial cells do interact with capillaries. The astrocytes have that direct interaction with the neurons, whereas the ependymal cells, they fill the hollow spaces in your brain and spinal cord with cerebral spinal fluid. So ultimately, they have two different targets. So that's the first two, and I would say the more difficult two. So now the next two for the central nervous system. Here's that term, oligodendrocytes, and we're seeing the word myelination. So earlier we just mentioned myelination is what gives the appearance of white matter in your brain or spinal cord, that lighter color, that's true. But truly the advantage is with proper myelination, the signaling of our neurons, the speed in which that electrical current travels the length of the axons is much, much faster. And a common kind of estimate around 290 miles per hour is how fast signal could travel through your nerves with that proper myelination. So you could quickly, in this case, with the oligodendrocytes specifically, you could signal from your brain down to your spinal cord, because keep in mind, it's just the central nervous system, you could quickly send a signal from your brain down to your spinal cord at a tremendous speed. But of course, there's also myelination in the peripheral nervous system as well. Oligodendrocytes, of course, unique to just the central nervous system myelination only. And then lastly, for the central nervous system, microglia, these are truly just the immune cells. So we'll just say the white blood cells, they're related to white blood cells. So they are unique to the central nervous system, and their job is to circulate through the central nervous system, brain and spinal cord, and respond to infection. So to try to fight off infection that's penetrated to the central nervous system. And so we have the first four here. So now for the peripheral nervous system, good news is we pretty much already covered this. Schwann cells are similar to oligodendrocytes, so meaning that they myelinate axons. So for that rapid speed, so I just mentioned the very rapid signaling through your nerves. In this case, for the peripheral nervous system. So think about the speed of signal traveling in the nerves of your arms with that rapid speed of Schwann cells. And satellite cells, similar to the astrocytes, but again, they're doing this job for the peripheral nervous system. So regulating the chemical environment surrounding nerve cells in the peripheral nervous system. So that'll finish up this section for chapter 13, the cells of the nervous system. Moving forward with the brain, a developed adult brain contains a large majority, so almost all of our body's neural or nervous tissue contained in just one organ. So that developed brain, that large mass. And in fact, again, the large mass, about 3 pounds, the weight for a developed brain. However, the brain itself is an extremely delicate organ. And the common description I give is it's really like the texture of firm tofu. So a living brain, or just freshly dissected, lacking any type of fixative, any preservative. It's a very, very delicate organ that, of course, is the control system for all of the other organ systems of our body. Starting out as early as 3 weeks into our embryonic development, the brain originally develops with these 3 primary areas here that are being highlighted. But ultimately, for lab, we're going to study the developed adult brain, and beginning with these 3 original primary vesicles, that'll lead on to forming now these 5 major areas in the adult brain. And again, it's these 5 areas here that'll be studied in more detail in the lab section. And we just see it spelled out here. So again, the 5 principal areas of a developed adult brain: telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. And just a quick look back at some of the terminology now. Gray matter, again, the clusters are principally neuron cell bodies, or in this case, nuclei. Most of them are found at the surface of the brain, but there is some deeper gray matter here highlighted in the various colors. So the deeper nuclei of the brain, whereas there's a large amount of white matter, myelinated axons, myelinated in this case by oligodendrocytes, neuroglial cells. And we also see that there's these hollow cavities inside of the brain as well. So there are some liquid-filled cavities, and these are all known as ventricles. There's the 4 primary ventricles of the brain, and there's also some smaller canals that connect the larger ventricles together as well. So the 4 major ventricles of the brain, and bordering on the edge of each of these ventricles, we notice here the ependymal neuroglial cells, the type of cell involved, of course, in producing cerebrospinal fluid. And we'll see definitely how these border each of the 4 ventricles. Will have their own groups of ependymal cells that then in turn produce CSF to fill and circulate the fluid through these various chambers. So taking a look at the fluid itself, it's another protective layer helping to support and protect the brain. So of course there's the bones of our cranium, there's what's called meninges, and there's also this fluid. And in fact, the fluid itself helps to support that 3-pound weight of the brain itself. So it's suspended in this fluid and supported by this fluid. And of course, since this liquid now has direct contact with the brain, with the nervous tissue, it's a way to deliver nutrients. So sugar, of course, oxygen to deliver that to the brain and to circulate the byproducts of metabolism and waste chemicals produced by the nerve cells and move that and flush that away from the brain. So this unique type of circulation, and it's found just in the central nervous system. Of course, the brain is the major part of the CNS. So to finish up with the ventricles, bordering each of those 4 principal ventricle chambers, this was called a choroid plexus. The term choroid always implies a rich blood supply. So we have a network of, in this case, capillaries. And this choroid plexus is involved in the production of cerebrospinal fluid. And of course, a major part of a choroid plexus, once again, are the ependymal neuroglial cells and the capillaries that they interact with. So it's that unit there, the ependyma and the capillaries together, that's what makes a choroid plexus. And again, this is now the exact structure that's creating the cerebrospinal fluid. Since we have the filtration of our cardiovascular system's plasma, it's filtered first through the ependymal cells, and that filtering of the plasma accounts for the differences between cerebrospinal fluid versus the plasma from our general circulation. And again, there's a choroid plexus, meaning that there's ependymal cells and capillary cells bordering each of the ventricles. So in the case of the lateral ventricles, which there's a pair, a right and left lateral ventricle, they're found on the inferior side of the lateral ventricles, whereas there's a choroid plexus on the superior edge of the third, as well as the fourth. And a look at that choroid plexus complex again. We can see the ependymal cells here labeled in pink. They're surrounding, you notice here, this capillary structure. And again, it's the plasma of the cardiovascular system that could pass through those pores. It now has to be filtered through the ependymal cell, and only the material that flows out the other side. This is now cerebrospinal fluid that's now entering and filling the various ventricles. So this diagram here shows the pathway of circulation, and we're not concerned with the actual steps and the arrows, but you notice starting from a choroid plexus of a particular ventricle, there's the production of the cerebrospinal fluid, and it does take a distinct pathway inside and around the brain, as well as you notice with the spinal cord. So again, the CSF is circulating through the entire central nervous system, brain and spinal cord, hence the name cerebrospinal fluid, brain and spinal cord. And ultimately, the capillaries of the choroid plexus, they're being supplied by, you notice right there, the carotid blood vessels that are traveling up the neck towards the brain specifically. And that's the source of plasma and blood supply for these choroid plexus regions. And to finish up this section now around the brain, discussing the layers of protection. So not only is there cerebral spinal fluid, the fluid cushion, but now there's also these physical layers of tissue called craniomeninges. These are found between the skull bones and the brain itself. And similar to cerebral spinal fluid, these meninges actually extend throughout the entire central nervous system. So for now, we're just focused on the brain specifically. But again, between the bones and the nervous system, we have these layers of meninges. And their purpose as a whole is to physically protect the brain, so absorbing any trauma to the brain, as well as just really stabilizing the position of the brain and maintaining that fixed position within the cranial cavity. So to start from the superficial end again, just directly deep to the bones, and in this case, the cranial bones, fused onto the deep internal surface of the cranial bones, we have this first major layer of the meninges known as the dura mater. And then deep to that, the second major layer, the arachnoid mater. And then lastly, the pia mater. This is now the layer of meningeal tissue that's directly connected to the brain and directly bonded onto the brain. So we have that order from superficial to the deep pia mater, which is ultimately connected to the brain. And here's just a nice picture that I really like. So taking a look back at several things at this point, we've already learned the skin, the integument, and then deep to the skin and any subcutaneous fat. We could get down to the muscles and, of course, the tendons. And aponeurosis, of course, the broad tendon. We discussed the bones. So, of course, the superficial membrane on a bone, the periosteum, and then the actual bone tissue deep to that. And we now see bonded on and fused to the inner surface of the cranial bones, that first layer of the meninges, dura mater, arachnoid mater, and then here we're still seeing the layer, a very thin, delicate layer, known as the pia mater. And here's just a view from one of the many cadaver textbooks. So again, we're seeing the area of the dura mater and a window cut through to see the arachnoid mater. So starting with the dura mater, the superficial layer, there are these distinct extensions, these large folds of the dura mater that do tuck much deeper into the cranial cavity. And an example, the falx cerebri versus the tentorium cerebelli are just a few examples. And these large ridges of dura mater extend into the deep fissures of the brain. And again, it helps to hold the position and maintain stability, in this case, between two different sides, two different hemispheres of the brain. And we'll see this in lab. So ultimately, here's a nice cutaway view, this coronal section. So we have, again, the dura mater. There's the arachnoid mater. And specifically, this deeper area known as subarachnoid space, it gets the name arachnid from that appearance of the collagen and elastic fibers. So collectively referred to as arachnoid trabeculae, just that network of connective tissue fibers. And that's, of course, what the meninges are composed of, layers of connective tissues. And another important trait about the subarachnoid space it's the area for circulation. So once again, we're seeing that cerebral spinal fluid. It circulates in the ventricles of the brain. And it also, as you notice here, circulates specifically in the subarachnoid space of the meninges as well. And then ultimately deep to the subarachnoid space, this thin, delicate layer directly bonded to the brain, and it follows every curve and contour of the brain tissue, known as the pyometer.